Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine if your own body held the key, not just to coping with, but actually fighting back against something like Alzheimer's. What if we could somehow, I don't know, supercharge the brain's own defense system, make it strong enough to clear out all that damaging stuff? Well, today, we're not just imagining. We're diving into some really groundbreaking preclinical work.
Yeah, it's pretty exciting stuff, just published in Nature Aging.
Right, and Cedars-Sinai highlighted it too. Researchers think they found, well, a surprising way to potentially treat Alzheimer's.
By boosting a specific enzyme. And right inside the brain's own immune cells.
Exactly. So our mission today, unpack this. Look at how they made the discovery, what it could mean for treating Alzheimer's.
And why this approach is causing such a stir, right?
Precisely. We want to give you that shortcut, those aha moments, straight from the cutting edge of neuroscience.
Let's get into it.
Okay, maybe just a quick refresher first. Alzheimer's. We know it's devastating. But at its core, it's about these two main things building up in the brain, right?
That's right. You've got the amyloid beta plaques, these sticky clumps outside the neurons.
And then the tau tangles inside the neurons.
Exactly. And together, they basically gum up the works. Communication breaks down, cells die, leading to memory loss, confusion, cognitive decline.
And the really tough part is there's still no cure. Over 7 million people just in the U.S. affected.
And that number's climbing, which is why research like this is so critical. We need something that doesn't just manage symptoms, but actually tackles the disease process.
So you mentioned the brain's immune cells earlier. Microglia, was it?
Yes, microglia. Think of them as the brain's dedicated cleanup crew, its own little innate immune system.
Okay, cleanup crew. So they're supposed to be clearing away debris, maybe even those amyloid plaques.
Ideally, yes. They patrol the brain, keeping things tidy, dealing with problems like protein clumps, crucial for brain health.
But in Alzheimer's, they drop the ball.
Ugh.
Or worse.
Well, that's the current thinking. They seem to get exhausted or maybe dysfunctional. They stop cleaning effectively, and sometimes they might even fuel inflammation, making things worse.
So if the cleanup crew isn't working properly, how do we fix that? How do we get them back on the job?
Well, that's where this study gets really interesting. It focuses on an enzyme called angiotensin-converting enzyme, or ACE.
ACE. Wait, that sounds familiar. Isn't that related to blood pressure? Like ACE inhibitors.
Exactly. That's what it's most known for, regulating blood pressure. That's why its role in Alzheimer's has been, well, a bit of a puzzle.
A puzzle how?
Because large genetic studies, these big genome-wide association studies, kept flagging the ACE gene as linked to the risk of late-onset Alzheimer's. But how it was involved wasn't clear.
So we knew it was there, somehow connected, but not what it was doing in the brain itself.
Pretty much. We mostly understood its job outside the brain. But this enzyme, ACE, it's what scientists call a promiscuous peptidase.
Promiscuous meaning?
Yeah, meaning it doesn't just stick to one job. It can chop up lots of different proteins, not just the blood pressure ones.
All right.
And critically, this new study highlights its ability to chop up the really nasty form of amyloid beta, the A42, into a less harmful version, A40.
Ah, so it's not just about blood pressure. It actually has this potential cleanup function directly on the amyloid itself because it's promiscuous.
Precisely. Its versatility is key here. It can act directly on those damaging proteins.
That really changes things. Okay, so how did the researchers actually test this? How do you supercharge just the microglia with ACE in like a living brain?
Very carefully, and with some clever genetics. They used a specific type of lab mouse called the 5xFAD mouse model.
5xFAD, what's special about them?
These mice are engineered to carry 5 human gene mutations that are linked to familial Alzheimer's. The result is they develop amyloid plaques and cognitive problems very quickly, much faster than humans.
So like an accelerated model of the disease. Makes sense for testing treatments.
Exactly. It lets researchers see effects much sooner. Then came the really precise part.
Which was?
They genetically engineered these mice so that only their microglia produced extra-human ACE. Not the neurons, not other brain cells, just the microglia.
Wow, that's specific. How did they manage that?
Using genetic tools to target the expression to those cells. They call these mice RACE+, R-A-C-E+. And they had control mice, RACE, without the extra ACE boost in microglia.
Okay, race plus with the boost, race without. Very targeted. So the big question then, did boosting ACE and just these microglia change anything? What were they hoping to see?
Their main hypothesis was, could this microglial ACE boost enhance their ability to deal with amyloid beta? And crucially, could that slow down or even reverse the neurodegeneration and memory problems in these mice?
They were aiming high then, not just tweaking, but really altering the disease course.
Absolutely, and the results were, well, pretty astonishing.
Don't leave us hanging. What did they find?
First off, a really dramatic reduction in amyloid plaques, all through the brain, but especially in key areas like the hippocampus, vital for memory.
Wow, so less plaque gunk.
Significantly less. And alongside that, they saw a shift in the amyloid itself, less of the really toxic A42 form compared to the less harmful A40.
Which suggests the ACE was actually doing that chopping job you mentioned, breaking down the bad stuff.
That's exactly what it points to.
Yeah.
More efficient processing and clearance of the harmful amyloid.
Okay, plaque reduction is huge. But what about the brain cells? Did the neurons survive better? Were the connections okay? Because that's where the real function happens,
You
right?
hit the nail on the head.
Yeah.
And yes, that's where it got even more compelling. The ARACE plus mice, the ones with the ACE boost,
Yeah.
they had significantly less neuron death, and maybe even more importantly, their synapses, the connections between neurons were rescued, back to levels you'd see in healthy mice.
Rescued, so not just protected, but brought back.
It looked like a reversal of that synaptic damage, which is a core feature of Alzheimer's, and the microglia themselves. They changed.
How so?
The ones with extra ACE became, let's say, supercharged. They clustered more around the plaques. They were much more aggressive in engulfing the amyloid beta.
Like they were really getting stuck in.
Exactly. And they seemed better equipped to actually break it down internally. Their internal recycling centers, these endolysosomal compartments, were enlarged and more active.
So the cells themselves transformed into more efficient cleaners. That's incredible.
Yeah.
But did it, actually make the mice smarter? Did their memory improve?
It did. That translation from cellular changes to actual behavior was clear.
How did they test that?
They used standard learning and memory tests. One is the Barnes maze, where mice have to learn and remember the location of an escape box. The RACE plus mice learned faster and remembered better. They found the box much quicker.
Okay, that's spatial memory. What else?
They also used fear conditioning tests. These assess memory for context and cues. The RACE plus mice showed significantly better memory recall in these tests too. Their freezing response, which indicates memory, was much stronger.
So real, measurable cognitive improvements. It wasn't just cleaning up plaques, it was restoring function.
That's what the data strongly suggests. Pretty remarkable.
It really is. You wonder, okay, how is this happening at the molecular level? What's ACE actually switching on inside these microglia to make them so effective? What's the mechanism?
Right. So they dug into the molecular details. They found ACE expression changed the activity of a whole set of genes in the microglia.
Including genes we already know are important in Alzheimer's.
Yes, like APOE and TREM2, major risk factor genes. ACE seemed to modulate their activity. But the really key finding seemed to be the activation of an internal signaling pathway called the spleen tyrosine kinase pathway, SYK.
SYK, okay, what does that do?
Think of SYK as a kind of central command switch. It drives the microglia's ability to move, engulf stuff like amyloid, and generally respond to their environment. It's crucial for their immune functions.
And ACE activates this SYK pathway.
It seems so. And they proved its importance really neatly. When they blocked SYK activity in the microglia that had the ACE boost.
Let me guess, the benefits disappeared.
Exactly. The enhanced amyloid cleanup just stopped. It showed that SYK activation was essential for that supercharging effect of ACE.
Wow. So ACE flips the SYK switch, and that gets the microglia into high gear.
That appears to be a major part of the story. They also saw other changes, like the microglia's internal trafficking system working better, and them producing more energy.
More fuel for the cleanup job.
Right, and genes involved in cell movement were upregulated, suggesting the microglia were better able to get to where they needed to be, like around the plaques. This whole coordinated upgrade.
It feels quite significant. Connecting the dots, this study seems to provide really strong evidence, at least in mice, that boosting ACE just in microglia could be powerfully protective.
I agree. It really shifts the perspective. It suggests we could potentially leverage the brain's own immune system in a very specific way.
And it flips the script on ACE inhibitors, doesn't it? Because some previous work suggested that reducing ACE might worsen Alzheimer's pathology.
That's a really important point. Yes, some studies hinted that inhibiting ACE systemically might not be good for Alzheimer's pathology. This work suggests that boosting it, specifically within microglia, is beneficial. It's about targeting the right cell type.
So what could this mean for actual human treatments? Are we talking about developing a drug that boosts ACE and microglia? Or something else?
Well, a drug is one possibility, though targeting it only to microglia might be tricky. What the researchers seem particularly excited about is the potential for a cell-based therapy.
Cell-based, like taking cells out, modifying them, and putting them back.
Sort of. The idea is you could potentially engineer microglia, maybe derived from patients' own stem cells, to express higher levels of ACE. Then, you'd deliver these supercharged microglia back into the brain.
Wow, that sounds futuristic. Is that feasible?
It's definitely advanced, but cell-based therapies are progressing rapidly. There are already clinical trials using engineered cells for other neurological diseases like ALS. So it's not science fiction anymore.
And why is that approach so promising here?
Because these ACE-boosted microglia seem able to overcome some of the problems caused by known Alzheimer's risk genes, like certain versions of APOE or TREM2. It's like providing a functional fix that bypasses the genetic susceptibility.
Like installing a software patch for the brain's immune system.
That's one way to think about it. It could potentially work even in people with high genetic risk.
Okay, the potential sounds enormous, but we need to be realistic. This was a mouse study. What are the big hurdles, the limitations we need to keep in mind?
Absolutely crucial point. The number one limitation is just that. It's mice. As Dr. Turtelot, one of the authors, emphasized, the absolute next step is confirming this works in human microglia.
Does human ACE do the same thing in human microglia? Does it activate SYK the same way?
Exactly. We need to verify that the fundamental biology translates. Human brain complexity is different. That's the critical translational work that needs to happen now.
What else? Any other unknowns from the study itself?
Well, they identified different subtypes of microglia, these disease-associated microglia or DAMs. ACE seemed to shift the balance between types A and B, but we don't fully understand what type B does or even if it exists in the same way in human Alzheimer's.
So more layers to peel back even within the microglial response.
Definitely. And understanding precisely how all the different affected genes and pathways contribute individually, cell movement, energy production, etc., will be important for refining any potential therapy.
Okay, so lots more research needed, but the core finding is incredibly promising.
I think so. It opens up a really novel therapeutic avenue.
So wrapping this up, this deep dive has shown us a really exciting new path, hasn't it? Harnessing the brain's own immune cells, boosting this ACE enzyme to clear plaques, protect neurons, maybe even restore function.
It points towards using our own biology in a smarter, more targeted way.
And it makes you think, doesn't it? If Alzheimer's might be tackled by precisely tuning our immune cells, what about other neurodegenerative diseases? Could similar personalized cell-based approaches work for Parkinson's or Huntington's?
It certainly raises that possibility. The idea of looking within our own bodies, using our own cells as therapeutic agents, is becoming increasingly powerful in medicine.
A fascinating thought to end on. The answers might lie within us, just waiting to be unlocked. We'll definitely be keeping an eye on where this research goes next, cell therapies, microglia. The deep dive definitely continues.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.